Okay, here we are looking at podcasts for Standard 2.1, and this is the first part of Standard 2.1. We're actually going to break this one up into a couple of different parts. So in Part A for this standard, we're going to look at the different cell parts and the organelles, and we're going to relate them to what it is that they do. So you'll be able to identify the different cell organelles, and you'll be able to describe the function for each of these organelles. Okay, so we're going to talk about a lot of different cell organelles, including things like the mitochondria, endoplasmic reticulum, Golgi apparatus, as we go through this set of notes. So before we get started, we're going to talk about what a cell actually is. When we talk about living things, all living things are made of cells, so cells is the basic unit of an organism. The discovery of the cell was made possible by the invention of the microscope. So people didn't know what cells were until the microscope was actually discovered, and then we started to figure out that we were made of these smaller little individually working units. So the history of the development of the cell and what we found. Robert Hooke is the first one who actually came up with the word cell. He looked at cork, which is basically dried plant cells, under a microscope, and he thought it looked like these cells that you would find in a monastery, which is the rooms all right up against each other. And so he's like, oh, those look like the cells that I would see in a monastery, and so he called them cells. About 10 years later, Anton von Leeuwenhoek looked at pond water and saw some living things actually swimming around in the pond water under the microscope, and that was something that nobody had ever seen before. And then a couple hundred years later, we had Schleiden concluded that all plants are made of cells. So he looked at a lot of different kinds of plants and noticed all of them were always contained different kind of cell-looking structures. The next year, Theodor Schwann decided to look at different animal tissues and also determined that all animals are made of cells. And then finally, Rudolf Virchow stated that all cells come from pre-existing cells that are already there, and that's basically the idea of the cell reproducing. So using all of this information in the history of the discovery of the cell, we have been able to come up with the cell theory. The cell theory has three parts. Number one, all organisms are made up of one or more cells. That is one of the characteristics of life. Some organisms are made up of only one, they're unicellular, and some organisms are made up of many, and those ones we call multicellular. Number two, the cell is the basic unit of structure and function for all organisms. That means that the cells make up what we are, and also the cells determine what we can do to stay alive, and they do that for all organisms. And the final part was that last thing that Virchow came up with, that all cells come from other cells that are already in existence. So all the cells that you have, you started out as one cell, and now you are made up of trillions and trillions of cells. So one cell divides into another, divides into another. So those are the main parts of the cell theory. Okay. We're going to concentrate on the second part of the cell theory in this one, and that is that the cells are the basic unit of structure and function for all organisms. So we're going to start taking a look at some of these terminologies. We're going to start with the fact that we are made of eukaryotic cells. A eukaryotic cell is a cell that has a nucleus and also has what's called membrane-bound organelles. That means that these are cell parts that are surrounded by a membrane. These are more complex types of cells, and we're going to take a look at the difference between eukaryotes and prokaryotes when we get to Standard 2.2. Now, an organelle is the functional unit of a cell. Organelle actually means little organs. So if you think about all your organs in your body and what they do for you, organelles have the same kind of thing going on within a cell. They allow the cell to do its job. Membrane-bound organelles, as I just said, means that they are surrounded by one or two phospholipid bilayers. So those ones are surrounded by these membranes. In our prokaryotic cells, the more simple ones, do not have membrane-bound organelles. Their organelles are much more simple. Okay, so what we're going to do is we're going to talk about the different organelles that are in the body. We're going to break them down by their overall function. Okay, so the organelles are going to allow cells to complete their jobs that an organism needs to do in order to stay alive. So a lot of different things that we have to be able to do to stay in homeostasis and stay alive. So the organelles of the body are going to allow us to do these things, like obtaining energy, protein synthesis. We have to be able to make our proteins, especially enzymes. Secretion and transport of molecules, so we have to be able to release things like hormones and whatnot. We have to be able to transport molecules in and out of cells and around the body, so our organelles are going to allow us to do this. Reproduction, we've got to be able to make more cells. If we can't make more cells, then when cells need to be repaired or they get sick, we can't fix them. Okay, so let's start to take a look at some of these different parts. We are going to categorize our organelles by function throughout these 2 podcasts. So we have organelles that act as boundaries. We have some that are going to act as controls. We have ones that are going to be doing assembly of the various organic molecules. Mostly, when we talk about this, we're going to be talking about proteins and lipids. Transporting molecules, we have to be able to transport stuff in and out of cells, around cells, and in and around the body. Storage, so you have certain organelles that are actually going to be involved with storing materials. You have our energy transformers. These are the ones that are going to allow us to get the energy that our cells need in order to do all of their various jobs. Support, there's a couple of different ways that cells are supported depending on whether you're talking about a plant or an animal. Locomotion, which is movement. Cells need to be able to move around, or they need to be able to move stuff within them. And so this locomotion piece falls in for both of those. And then we have some cell parts that are used exclusively during the process of cell division. Okay, so we're going to get started with boundaries. The number 1 boundary for all cells is the plasma membrane. The plasma membrane is also called a cell membrane, so you will see that used either way. Sometimes it's called plasma membrane, sometimes it's called cell membrane. Your plasma membrane serves as a boundary between the cell and its external environment. So basically it's the outer walls of the cell. Everything gets held inside, and it keeps all the other stuff out that is not supposed to be in there. So the cell membrane, the plasma membrane, is selectively permeable, so it's going to allow certain materials to pass in and out of the cell, but not just anything. So it's going to be a highly controlled process. And again, all cells have this membrane surrounding them to control what is going in and out. Your cell membrane is actually holding in also all of the cytoplasm. Another boundary that some cells have is a cell wall. Cell walls are only found in plants, bacteria, fungi, and some of our protists that are out there. So we'll get into the different categories of living things in a later time. But animals like us do not have cell walls surrounding any of our cells. But these types of organisms do. It goes right around the outside of the plasma membrane, and it's going to allow for extra support for the cell and also extra protection for the cell. Animals tend not to need that because we have other ways of protection, the way that our cells are put together, and also we have immune systems and transport systems and stuff, so we don't need to have these cell walls. If we did, then our skin wouldn't be flexible. It would be hard like bark on a tree, and that would not be a really pleasant way to move around. Okay. The cell wall is also selectively permeable, just like the cell membrane, so it is going to help to select which things can go in and out of the cell. Okay. Controls are up next. Your main control for your cell is the nucleus. Nucleus is going to regulate all of the different cell functions for the body. It is surrounded by a double-layered membrane called a nuclear envelope. And in that nuclear envelope, there's actually large pores that are going to allow materials to pass in and out of the nucleus. So you're going to get large amounts of things going in and out of that nucleus. So there's big holes that are in the membrane of the nucleus. Inside the nucleus, you also have chromatin, which are your long tangles of DNA. Remember, DNA is your genetic material. So all your genetic material controls everything that your cells do. And so all of that stuff is inside of the nucleus. Okay. Also with controls, part of the nucleus at the center of it is a structure called the nucleolus. Okay, this is found inside the nucleus, and its job is to make ribosomes. Okay, that's the main job of the nucleolus, is to make ribosomes for the cell. Ribosomes are going to have a very important job that we'll talk about in just a moment. Okay, we'll talk about right now. Ribosomes are made for proteins. So ribosomes are going to be making proteins for the cell. That is their job. That is what they need to do. And so the nucleolus is going to make those ribosomes for us. Okay, continuing on with controls, we have our chromosomes, which are inside of the cells. These are basically highly condensed chromatin. So when your cell is relaxed, your DNA is actually a big blobby mess like that. But you can't really see that. So when we can see our DNA, it's in a condensed form like these chromosomes. So a chromosome is actually composed of two identical chromatids. It's got these two arms on them that are attached to each other in the middle. Okay, and basically what you have is your DNA that was all loose like this is now tightly wrapped around some proteins, and it makes this really tight structure. This is actually going to be useful when the cell divides. Okay, so here's what our nucleus will look like. So inside is the nucleolus where the ribosome gets made. Covering all that, all in here, are the chromatin. That's the natural state of your DNA, nice and loose. And then around the outside you have your nuclear envelope, and you can see that there's little holes in there, and those little holes are the pores where RNA molecules can actually get in and out of the nucleus. And then attached to that are a couple of other structures that we're going to be talking about. Okay. So what the cell is actually controlling is the assembly of different molecules. Most assembly happens in the cytoplasm of the cell. This is jelly-like material that surrounds all of the organelles inside of the cell. And it is mostly water. And it is the site of chemical reactions within the cell. So all the different kinds of chemical reactions that have to happen for the cell, like making proteins and breaking things down, all that kind of stuff is going to happen in the cytoplasm. All of our salts and everything are dissolved in water in the cytoplasm. So it's mostly water in there, and then the organelles are mixed in there. Okay. Now, also in there, certain ones that are in there, ribosomes, are a very important organelle that are inside the cytoplasm. These are small organelles that are made of RNA and proteins, and their job is to make proteins. Okay, this is where your proteins get made. And remember, proteins determine everything about you. So without your ribosomes, you wouldn't make proteins, you wouldn't exist. So ribosomes are actually one of the few organelles that all living things have, because all living things have to be able to make proteins in order to survive. So some of them are found floating free in the cytoplasm, and they're going to make proteins that are going to be used by the cell. So the ones that are floating free are going to make things that are going to get used by the cell. You have others that are attached to another structure called the rough endoplasmic reticulum. Those proteins that get made there are actually going to be exported from the cell. Those are the ones that we could use in our body, like different hormones and things like that. Okay, so also helping with assembly is the endoplasmic reticulum. There's two types of endoplasmic reticulum. Smooth ER, main job is to make lipids. Okay, so these lipids could become our steroids, could become our cell membranes. But the smooth ER's main job is to make different lipids that the cells in our bodies need. Okay. We're going to talk about the rough ER here because the rough ER is a delivery system. So smooth ER is going to be making proteins, but the other ER, which is our rough ER, is actually a folded membrane, and it's going to be a delivery system. So the rough ER is studded with those ribosomes, and that's where we're going to make all of our proteins. And once those proteins get made, they're going to get transported to different parts of the cell. Okay, so here's the nucleus, and you have the rough ER is attached directly to it, and those are the little ribosomes on there that are going to make the proteins. And then sometimes the smooth ER is actually a continuation of that. Sometimes this will be a separate structure all on its own. But the proteins basically get made by the ribosomes, and they're going to travel through this rough ER as they get processed. Also used for transport is the Golgi apparatus, which is sometimes called the Golgi body. Okay, this is a series of flattened stacks where the new lipids and proteins that just got made in the endoplasmic reticulum are going to get packaged up and shipped. And they're going to get shipped out to the plasma membrane, and then they can get sent out of the cell. So they're kind of putting the finishing touches on the proteins and lipids that get made before they go off to their final destination so they can do their jobs correctly. So your Golgi apparatus actually almost looks like a stack of pancakes, these folded membranes, and then coming off of those membranes you have these things. This is a vesicle, and that's a little transport molecule or transport structure inside the cell. So whatever was made is now inside of that vesicle, and that vesicle will travel over to the side of the cell and drop that stuff off. Okay, so all of this comes down to making a new protein. So when we're going to make a protein, the first thing it starts with is DNA. We have DNA that's in our nucleus. The DNA is going to use RNA, and it's going to send a message out to the ribosomes. Okay. The RNA is going to attach to the ribosomes on the rough endoplasmic reticulum, so on the rough ER. The RNA leaves the nucleus, goes over to the ribosomes on the rough ER. There, the proteins that have been made are then going to be transferred over to the Golgi apparatus. Okay, so DNA has the message, gives it to RNA. RNA tells the ribosomes, you need to make this. Endoplasmic reticulum sends it to the Golgi apparatus. The Golgi modifies and refines those proteins, finishes them off, puts on any finishing touches. And then the vesicles that the Golgi makes are going to bring those proteins over to the cell membrane, and they will get exported from the cell. So in the second podcast, we're going to take a look at the rest of the organelles that make up the eukaryotic cells.